The overall goal of the following experiment is to use the fluorescent proteins morange2 and DRONPA for dual probe optical highlighting to selectively label 4 individual cell populations or cellular compartments. This is achieved by first illuminating cells with low-intensity 488 nanometer excitation light to switch off all the green DRONPA fluorescence. As a second step, cells are exposed to high-intensity 488 nanometer excitation light, which photoconverts the orange fluorescence to red. Next, cells are exposed to 800 nanometer 2-photon excitation in order to switch on DRONPA fluorescence again. Results are obtained that show it's possible to label cells in 4 distinct combinations based on dual probe optical highlighting with morange2 and DRONPA.
The main advantage of this procedure over existing experiments like subcellular fractionation is that it's done on live cells. This procedure can help answer key questions in beta cell biology, such as why only a small fraction of insulin vesicles are secreted. Although this procedure can provide insight into beta cell function, it can also be applied to other systems, for example, tracking cancer cells in vivo.
A fluorescent protein droplet sample consists of an octanol water emulsion with the fluorescent protein residing in the water phase. This emulsion is sandwiched between a microscope slide and a 22 mm square cover glass for microscopy applications. At the beginning of this procedure, the microscope slides and cover glasses must be cleaned and coated with a hydrophobic agent. Clean the glassware by washing it for 5 minutes with acetone. After washing, leave the glassware to dry by air. Next, prepare a 2% methyl trimethoxysilane solution in acetone and coat the glassware for 2 minutes in the solution. After coating, remove the glassware from the solution and allow it to dry. Then rinse the glassware with 70% ethanol from a spray bottle. After rinsing, let the glassware air dry again and remove any remaining liquid using pressurized air. The coated glassware can be stored for at least 1 month. Once the glassware has been prepared, obtain fluorescent proteins that have been previously purified as His-tagged protein from E. coli. Measure the absorbance spectrum of the purified protein. Prepare a stock dilution with an optical density of approximately 0.1 in tris buffered saline solution with pH 8 containing 0.1% bovine serum albumin. Additionally, prepare 10 ml of a 1:1 mixture of 1 octanol and tris buffered saline solution in a 15 ml conical tube and mix vigorously. After mixing, leave the mixture until the phase separation is complete. The top phase is the octanol. To make the emulsion, pipette 45 microliters of 1 octanol and 5 microliters of fluorescent protein into an Eppendorf tube. Tap the tube a few times to start formation of the emulsion. Then, sonicate the tube for 30 seconds in a sonication bath. Meanwhile, get a coated microscope slide and cover glass ready. Following sonication, the emulsion should be completely cloudy. Immediately pipette 4 microliters of the emulsion from the middle of the tube onto a coated microscope slide and cover it with a coated cover glass. The emulsion should spread evenly between the microscope slide and the cover glass. Within minutes, the sample should be stable. The following procedure is a general strategy for setting up a fluorescent protein photoconversion experiment on a confocal laser scanning microscope. This procedure can be applied for purified proteins as well as for live cells. In this experiment, a droplet sample containing purified morange2 protein is used. As a general starting point for the photoconversion experiment, set up the confocal laser scanning microscope as follows. Use a 40x 1.3 numerical aperture oil immersion objective and an image size of 512 by 512 pixels. Set pixel dwell time to 6 microseconds. The scan zoom to 4 and increase the pinhole size to match a Z resolution of 3 microns. Configure 2 detection channels for the initial and photoconverted fluorescence, as well as a photoconversion channel. Observe the orange species using 561 nanometer excitation light and collect the fluorescence between 570 and 630 nanometers. Detect the photoconverted red species using 633 nanometer excitation light and collect the fluorescence between 640 and 700 nanometers. For the photoconversion channel, select the 488 nanometer laser line and collect the fluorescence between 490 and 540 nanometers. Imaging the photoconversion channel is not strictly necessary, but can be helpful. To begin the experiment, use the detector channel to image the initial fluorescence with continuous scanning to adjust the laser power and detector gain for optimal image quality. Then, activate the photoconversion channel and select a low laser power. Start imaging a time-lapse series and gradually increase the photoconversion laser until significant bleaching of the initial fluorescence is observed. Continue scanning until the initial fluorescence is approximately 75% bleached. Next, deactivate the photoconversion channel and activate the detection channel for the photoconverted fluorescence. Start imaging with a high detector gain and low laser power. Then, gradually increase the laser power until the photoconverted fluorescence is detected. Once detected, adjust the laser power and detector gain for optimal image quality. Finally, optimize the laser power used for photoconversion as well as its duration. Increasing the laser power will accelerate the rate of photoconversion. However, too much laser power will photobleach the protein. Once the optimal photoconversion laser power and duration have been determined, these parameters can be used to configure a standard photobleaching module or fluorescence recovery after photobleaching module. Then the photoconversion channel will no longer be required. Due to the red-shifted spectral properties of M-Orange-2, it can be used in combination with the green photoswitchable fluorescent protein DRONPA for dual probe optical highlighting. To begin dual probe optical highlighting, grow HeLa cells in glass bottom MatTek dishes. Transfect the cells with plasmids encoding the fluorescent proteins 24 hours prior to imaging using a standard lipofectamine 2000 transfection. Place the live cells on a confocal laser scanning microscope and set up the microscope for M-Orange-2 photoconversion as described earlier. Then, configure the microscope for DRONPA photoswitching. DRONPA is excited with 488 nanometer light and its fluorescence can be imaged using the M-Orange-2 photoconversion channel. Be sure to minimize the laser power used for imaging DRONPA, as too much laser power will cause inactivation of DRONPA. Add a channel for DRONPA photoactivation using 800 nanometer 2-photon excitation. Subsequently, determine the laser power required for imaging, photoactivation, and photoinactivation of DRONPA fluorescence.
One of the most difficult aspects of this procedure is to inactivate DRONPA without photoconverting M-Orange, because both events take place at the same wavelength. In order to prevent this, it is very important to carefully optimize the photoconversion laser power and duration.
Take caution as photoconversion of M-Orange-2 and inactivation of DRONPA both occur upon excitation with 488 nanometer light. To inactivate DRONPA without significant M-Orange-2 photoconversion, use a lower laser power. Once the parameters for M-Orange-2 photoconversion and DRONPA photoswitching are set, dual probe optical highlighting can be achieved. First, inactivate DRONPA fluorescence in the whole field of view with low power 488 nanometer excitation. Then, select a region of interest and photoconvert M-Orange-2 with high power 488 nanometer excitation. Finally, select a region of interest and activate DRONPA fluorescence using 800 nanometer 2-photon excitation. Fluorescent protein droplet samples are ideally suited for setting up confocal microscope photoconversion applications. When the droplet sample is prepared correctly, the droplets appear very homogeneous. However, when the sample is prepared without coating the microscope slide in cover glass, the droplets tend to be squashed against one of the glass surfaces. In samples prepared in the absence of 0.1% BSA, the fluorescent protein tends to accumulate at the 1-octanol water interface, creating a halo of fluorescence. Photoconversion is critically dependent on the laser power and the duration of exposure. Too much laser power or too long exposure will cause photobleaching and results in a reduction of the photoconverted red fluorescence. The distinct spectral properties of M-Orange-2 and DRONPA permit dual probe optical highlighting. Prior to photoconversion, cells expressing M-orange2 histone H2B and DRONPA mito show orange fluorescence in the nucleus and green fluorescence in the mitochondria. When DRONPA fluorescence is switched off with low-power 488 nanometer excitation, there is minimal photoconversion of M-orange2. M-orange2 can then be photoconverted to red with high-power 488 nanometer excitation. Once M-orange2 has been photoconverted, DRONPA fluorescence can be switched back on using 800 nanometer 2-photon excitation. With dual-probe optical highlighting, cells can be labeled in 4 distinct ways. Using this technique, it is possible to specifically label 4 different cell populations.
As you've seen, one of the most difficult aspects of this procedure is to inactivate DRONPA without photoconverting M-orange. Once mastered, further experiments can be performed using this system. For example, tracking multiple vesicle populations in pancreatic beta cells. So that's it. I hope you found this presentation useful, and good luck with your experiments.